Hi, my name is April, and the protein that I decided to make a model for is Piezo1. So Piezo1 in the middle here is a protein that functions as a mechanosensitive ion channel. It's stimulated by changes in membrane tissue, so induced by things like mechanical stress, which causes this channel to open and allow the flow through of cations such as calcium. This is going to be embedded in the phospholipid bilayers and plasma membranes of many different types of cells. When you have a gain of function mutation of Piezo1, you can get a condition called dehydrated stomatocytosis, DSH, which is also known as xerocytosis. And this is caused by gain of function mutation, which causes excessive calcium intake, leading to ionic imbalance and dehydration of red blood cells. So for my model, I chose to represent the phospholipid bilayer that Piezo1 is embedded in using cotton balls for the phosphate heads and toothpicks for the nonpolar fatty acid tails. I used cardboard to create the foundation for the protein and overlaid it with tissue paper. I represented the calcium ions flowing through using orange colored cotton balls. Overall, this structure of Piezo1 is a trimeric propeller-shaped structure with 3 peripheral blades and a central pore. So this is the protein contains 3 separate subunits that can be visualized in the aerial view, but I decided to show it in a side view so that you were able to see its location within the phospholipid bilayer and its function as a ion channel. So onto the structure. So Piezo1 contains up to quaternary structure. So the primary structure is the long chain of amino acid sequences. The secondary structure is the polypeptide backbone that includes alpha helices and beta sheets. The tertiary structure is a single subunit of a peripheral blade, and the quaternary structure is all 3 peripheral blades. My model shows the side view where you can see 2 of the peripheral blades that make up the quaternary structure. So our assigned mutation for Piezo1 was an arginine replaced by a histidine at amino acid 2456. So while this mutation directly affects the primary amino acid sequence, the protein structure does not appear to be very different at higher order structures. The mutation's increased calcium influx is represented by this pink. These are also calcium, but they're adding to the whole supply of these orange cotton balls, which also represent calcium. The point mutation we were assigned impacts the protein's function by making the channel more active and sensitive. So the channel is able to open easier and stays open for longer durations, which allows more calcium to flow in. This eventually is going to dehydrate red blood cells, and this is the condition DSH. So the mutated amino acid is located pretty far along in position 2, 4, 5, 6 of the primary amino acid sequence. My model is showing a higher order structure than the primary sequence, so you can't really see the exact change of the amino acid, but it is showing the effects of what that point mutation would do when you convert an arginine for a histidine. So this model obviously comes with a few limitations because it's made with household materials. The first is that the phospholipid membrane that's represented by these toothpicks and these cotton balls are static. They don't move, and so it doesn't reflect a real life scenario where you would have mechanical stressors that would lead to the curving and flattening of the phospholipid membrane. And so it is in this structure, it's just going to appear linear, even though this is not the case. The second limitation is that this is a side view of Piezo1, so you can't really see the full trimeric blade-like structure that an aerial view would show, but this does also have its advantages as kind of showing the location and its function better. And the third limitation is that the Piezo1 is displayed by cardboard, and so you can't move it and show it closing and opening. So this kind of makes it appear as if it's always open, constitutively open, which is not the case. It is actually opening and closing in response to mechanical stimulation. Yeah, thank you.